The next speaker is Vera Komeyer from Research Center Jülich in Germany.
Hello everyone. In the next 10 minutes I want to introduce some methods and how we can arrive at proper selection of adjustment variables to deconfound or debias predictive models. Oh, it doesn't work. I just have to press this button, right? Okay. I have no disclosures. First of all, the question is, why do we even want to use predictive models? And usually we either want to uncover neurobiological mechanisms, or we want predictive models as clinical tools to then use them in a clinical setting. For both of these scenarios, we need reliable predictive models that generalize across settings. The problem, however, is, and I think most of you are aware of this problem, is that we have confounding variables that introduce spurious association and thereby mask the association of interest between our brain features and our behavioral target. Oftentimes, confounders are defined as any kind of third variable that correlates with the features and the target, but are considered irrelevant for the research purpose. In this talk, I want to elucidate that the correlation definition given here is not enough, and I will introduce a three-step approach in how you can determine which variables you should actually correct for in your predictive modeling approach. And I also want to elucidate a bit on the question if deconfounded models could support causal claims. To start off, we first need to consider different types of third variables. And most of you are maybe aware of confounding, but we need to distinguish between different types of third variables. And confounders, in a proper definition, beyond the correlative definition, need cause-effect definitions. So a confounder is defined as a variable that affects both the feature and the target. And one could, for example, have a look at a more applied example here. When you're interested in the connection between hippocampal volume and depression, then early childhood trauma would affect both of these variables, and then you should correct for this variable. However, we have other types of third variables, like colliders, and you see already that the arrows point in the opposite direction. So a collider is defined as a variable that is basically affected by both the features and the target. An example could be if you're interested in the association between serotonin receptor function and cortisol. They both affect depressive symptoms as well. However, if you control for depression in that case, you would introduce bias into your model. So you should not correct for a collider. And the third type I want to introduce is a mediator, and I think most of you are maybe familiar with mediators as well. And mediator basically is just a detour route connection between the feature and the target. For example, if you're interested in the association between amygdala hyperactivity and depression, you also have this detour via cortisol. And if you correct for SET here in this case, then you get only the partial direct effect. The problem now, as introduced before, is that all of these types of third variables produce the same correlation between the third variable and the feature and the target. And that's why the initially introduced correlative definition is not enough, because then we might end up with adjusting for a collider and thereby introduce bias, which is a problem. To overcome this, we need to use the causal definition as I just introduced, and so-called directed acyclic graphs are very handy there. And the little arrow pictures you saw before is already a small version of a DAG, basically. So in the three-step approach that I want to introduce, the first step is that you can now create a DAG around your research question of interest. And you do that by asking what are known or conceivable causes of the target. And you continue this question bottom up by recursively then adding variables and asking for each added variable what could be known or conceivable causes of that variable. And you answer the question based on domain knowledge and existing literature. And you stop when a sufficient set of deconfounders is identified. Let's have a look at an example. If you were interested in gray matter volume features as our brain features and hand grip strength as a target, then this could be one example DAG for this research question. So we could ask what are known and conceivable causes of grip strength, and then sex will influence grip strength. I think that's very obvious. Or muscle mass would influence that. And then you would ask what are known or conceivable causes of muscle mass, and then sex hormones or age would be causes of that again. And by that you build bottom up a DAG for your research question at hand. Importantly, this causal analysis can lead to different DAGs because it's of course to some degree subjective and not data-driven, because you cannot do that in a data-driven way. But still it allows for transparent communication of your assumptions, and that's why it's a very handy tool. Now the question is, what does it mean that you have a sufficient set of deconfounders? And that's where the second step comes in. And one option to identify a proper set of deconfounding variables is the so-called backdoor criterion, which uses backdoor paths in the dark, which are paths that go from X to Y that start with an arrow pointing into X. If you have a look again at our confounder, small little DAG here, you see that there's an arrow incoming to X from Z, and you want to block all pathways that start off like that. As you noticed, I used here the word deconfounding, and confounders can differ from deconfounders because deconfounders are a sufficient subset of confounders whose adjustment blocks all confounding pathways. For example, you could have a confounder in your little DAG, which is unmeasured, but then you have on this non-causal pathway, remember that's the pathways with incoming arrows into X, you might have a deconfounding variable that you have measured, and if you then adjust for the deconfounder, you also block this pathway even though you have not actively measured your confounder. If you have a look at our example DAG here, I just highlight 2 example confounding pathways here. There's many more in this DAG, but just to not get it too crowded. And one would lead from gray matter volume over sex hormones muscle mass to hand group strength. In another example, one would lead over age, sex hormone sex to muscle mass. If you now apply our backdoor criterion, a first set of proper deconfounding variables would be sex hormones and age. However, assume we don't have sex hormones measured in the data set we are using. Then we can make use of the concept of the deconfounders, and a second set of proper deconfounders to adjust for would in this case then be sex and muscle mass. In the third step, after we causally identified a proper set of deconfounders, in our case now sex and muscle mass, we can basically adjust for this variable blocking these pathways. There's more options to do that beyond the backdoor criterion, and if you're interested in that, you can have a look at my poster and we can discuss about further options there. Once you identified the variables, you still need to statistically evaluate the relationship between the identified deconfounders and your features on your target, because you need to see if there's shared signal components there, of course. And in our case, we see that this is the case. And additionally, it would be good if you have also a look at the statistical correlations between your feature and your target to see if your analysis makes sense to begin with. And then if we take this example again and we make an unadjusted prediction, in this case just a linear support vector regression, we see that we have a high correlation between the true and predicted target, in our case hand group strength, and this is now the unadjusted model in an out-of-sample prediction. However, this good performance we get here, it's the biased model. Why do I know this is biased? Because if I now adjust by linear feature residualization for the identified deconfounders, we see that in the unbiased model or debiased model, the performance is a lot lower. And now everyone might think, okay, but now I have a model that has a low performance, and what is this model useful for? Well, it's still biologically more meaningful because we learned something about the underlying biology. Because what we did by adjusting for these confounders, we blocked this spurious association, and the debiased model just gives us information about the association of interest that we were interested in to begin with. So by debiasing the model, we learned, okay, maybe the linear prediction model was not enough, or I would need different brain features. And by that, I already learned something about the biology, while the model depicted in purple has a high performance, but biologically, it's meaningful to derive any insights about what's happening in the brain, basically. I want to highlight that the linear feature residualization I used here for this example has its limitations, but it's widely used in the literature and neuroimaging predictive modeling literature. But obviously, we use a linear model for adjustment, so there might be residual nonlinear signal that might be picked up by a more complex algorithm. And also, usually only features or targets are residualized, so we might have like leftover confounding signal in the respectively other one. There is options out there, for example, the double debiased machine learning approach as suggested by Janosok 2018, but this is meant for treatment outcome modeling, and to adapt it to the supervised machine learning case, it needs further modifications, which I don't want to go into depth right now here, but if you're interested, we can also discuss it at my poster. So now coming to the second questions, if we have now deconfounded our models very properly, might they allow for causal insights? Well, let's take one step back. Traditional machine learning, as we do it, learns probability distributions, so probability of y given x. There is causal machine learning approaches out there, however, they aim to estimate the treatment effect on an outcome. So the better question we would want to ask maybe is not if they allow for causal insights in the sense of treatment outcome effects, but more but more in the sense of understanding causal mechanisms between features and targets. And the short answer is no, they do not directly, because there's further causality assumptions that need to be fulfilled. And additionally, we need to take care of the data generation process, because there can be effects like causal and anti-causal learning. So what should you take from this little presentation? First of all, do not blindly adjust for any type of third variable or just for default ones like sex and age, but build a DAG around the research question you have. Second, linear feature residualization comes with limitations, but alternatives need further methodological adjustments. Deconfounded models allow for biological, more meaningful insights, even though predictive performances might be lower, but causal claims can and should only be made under intense constraints. And lastly, justifying your confound adjustment through causal tools offers the opportunity to think about relevant relationships, to communicate assumptions transparently, and to identify variables that you should integrate, and also, maybe even more importantly, that you should not integrate, to not introduce inadvertently collider bias. And with that, I thank you for your attention, and all my supervisors and group.
There is one question, no, I think now two already, and a couple. So I start with the first one now. Can we have bidirectional arrows in the DAG, or bidirectional effects, or unclear causal directions? If yes, how to handle them?
Yeah, that's actually a very interesting question. So, of course, if we use a DAG, the ideas, I mean, the difference between a causal and a correlative depiction of a pattern is that you have an arrow in one direction or a bidirectional arrow, where the bidirectional one would be the correlative associational one, and the one directional arrow would be the causal assumption, basically. And so if you build this DAG, you would always want to have one directional arrows, because you really put causal assumptions in there. However, of course, in a field like neuroimaging, where you have a really interwoven relationship between many variables, it can be that you have circular errors, like even via some other subvariables then. And this is actually a problem, because of course it's directed acyclic graphs, so you assume that there's no cycles. And this problem of potential cycles in the DAG is really a problem in our field. And I right now, unfortunately, don't have a direct question to this, but I think this is quite an ongoing field of research, actually.
Okay. There are many questions, okay. So perhaps people can come and talk to you at the end. I will make a quick one. How to differentiate mediation and confounding? Actually, how easy is to do this DAG? How easy is how to differentiate mediation and confounding, and how easy is to build the DAG? Because in some situations, it might not be very easy to build.
Yeah, exactly. So the differentiation between a confounder and a mediator is, maybe you remember the first little diagram I showed, basically either going with the arrows in the same direction, basically, or where you have this little detour. And then building the DAG, basically, I think the best approach is if you go really like in this bottom-up manner I presented, that you recursively or iteratively build it really bottom-up to then see, okay, when do I have to stop and not be overwhelmed by a million variables you don't know how to put in, but be really stepwise ask for each variable precisely about the connections it has with other variables. But I'm happy to discuss it also further at my post.
Yeah, there are many questions.
Okay.
I think.
Go on.
Yeah,
with
go.
this.
Yeah.
Thank you.
Thank you very much.